the gift of threonine. Superficially, it looks like valine. They both have that kind of V structure, but that's pretty much where the similarity stops. You see, threonine has an OH group, it has a hydroxyl group, an alcohol group, whatever you want to call it. It has a functional group that valine does not. Valine's just got like pure hydrocarbons in its side chain. Boring. Now we're introducing some fun things. In this case, a hydroxyl group. That hydroxyl group is going to do a few things. One is it introduces polarity. The oxygen pulls on those electrons that it's sharing with the hydrogens, with the carbons, and so it's going to make that oxygen partly negative. This is going to give it partly negative and partly positive parts. So the hydrogen is going to be partly positive, the oxygen is going to be partly negative, and this is going to make it so that it's going to be polar, unlike valine, which was going to be nonpolar. So we pulled this over to the polar category, which is going to make it so that it's a lot less hydrophobic than valine. And so you're more likely then to be able to find it multiple places in the protein, including on the surface, on the inside, kind of still, because it still has those hydrophobic parts. So it's kind of in the middle, but it's a lot more flexible that way. But speaking of flexibility, both threonine and valine are what we call branched chain amino acids, and they have their branching close to their backbone. And this is going to make it so that it's hard to rotate around them. And so, or without getting like clashes and things hitting one another. And therefore, you're not very likely to find it in an alpha helix, but you could find it in a beta strand. That OH group, that oxygen, it's what we call a nucleophile. It's a nucleophile. It loves nuclei because nuclei, they've got protons. And so it's going to go and attack electrophiles. Electrophiles are things that want electrons, and the nucleophiles are things that want to give away those electrons, or at least share the electrons with something. So what happens is that this threonine, this OH, not only does it polar, but it's also nucleophilic, and it can go and it can attack a phosphoryl group on ATP. And this is going to then make it so that you get what we call phosphotyrosine. And so this is an example of a post-translational modification, a type of modification to a protein that gets made after the protein is made. And then this can change the shape of the protein. It can cause the protein to maybe activate sometimes. It can add a new binding site. Lots of cool things that can come from the phosphorylation. And phosphorylation can only happen at certain sites. And so typically it's going to be like tyrosine, threonine, and serine are the three main ones. Kinases are the enzymes that put the phosphate group on. Or I guess it's technically a phosphoryl group. Technical note I'm not going to go into here, but I will say that you often hear of like serine threonine kinases. So different kinases will recognize different sequences and phosphorylate them. And often kinases will be able to kind of recognize similar sequences, but not exactly the same. So we refer to like a consensus sequence, which is like, okay, they'll phosphorylate this residue. So basically the leftovers of the amino acid, when you hook them up, we call them residues. So we can refer to like this is in a chain, this is now a residue. And so this would be a serine, this would be a threonine residue. And then when you phosphorylate it, well, now you have a phosphothreonine residue. And that was being put on after you already made the whole protein. If we go and we were to say, okay, well, how does the kinase know what to phosphorylate? It would have sequences on either side of that residue that the kinase likes. And so it would kind of complement the binding side of that kinase, and the kinase would then phosphorylate it. Because serine and threonine are both kind of small, the kinases can often phosphorylate both of them. And so you'll talk about like serine threonine kinases that'll be able to phosphorylate both serine and threonine. And then you'll think about like tyrosine kinases. Those are typically different kinases because tyrosine's all big and bulky. Tyrosine kinases, you often hear about them in terms of receptor tyrosine kinases, which are important regulatory molecules that are able to pass signals down into cells. An example of this is the insulin receptor. That's sort of the biochemical stuff to know about threonine. It's polar, it's got that nucleophile, it gets phosphorylated sometimes, and the kinases do that. It's branched, and the branching is close to its backbone, so it doesn't want to be in alpha helices. And it's still not too polar, but it is polar because it has that OH group. I like to remember the name as like 309. Like it's got three like kind of groups off of its end, and one of them is an oxygen, so O. That's a helpful mnemonic to remember the shape of threonine, but it's not the actual way that it got the name. Instead, the name comes from its structural relationship to the four-carbon carbohydrate threnodic acid, which itself got its name from the sugar threose, which it got its name from the scrambling up the letters of a version of erythrose. And then erythros got its name because it turns red in basic solutions. Kind of went down a little etymological rabbit hole, chemtomology.co.uk, which I recommend if you are a geek like me and want to know where words come from. Threonine is essential in a dietary sense, so we need all of these amino acids, but some of them we can make ourselves. Threonine is not one that we can make ourselves, and so instead, we are going to actually have to get it through our diet. And we know this thanks to William C. Rose, who also discovered threonine. And so I want to tell you about his work. How did he find threonine? Well, he did a lot of work. It took 40 kilograms of initial protein that he started with, and he ended up with only 4.5 grams of threonine. And he went through a lot of stuff. So I'll walk you through some of the main things from his paper, as well as then some work that he did where with grad students to figure out it was essential. Threonine was actually the last of the kind of main 20 amino acids to be discovered, and this is going to be happening in 1936. Rose is really interested in figuring out which of the amino acids are essential. So which of the amino acids do we need to actually get in our diet, and how much of each of them, and how many amino acids are there, and all this cool stuff. He wasn't the first person to be interested in this. Instead, he was building upon the work of previous scientists. In the 1910s, a couple of scientists, Osborne and Mendel, fed rats diets with restricted protein sources, and they found that the rats fed protein sources that were deficient in lysine would stop growing unless you added back lysine. And similarly with tryptophan. And so that was how they found out that tryptophan and lysine were essential. They were able to do this without actually purifying each of the amino acids, just kind of giving it a protein source that didn't happen to have all of the amino acids in it. Then in the 1930s, however, the American biochemist William C. Rose decided to do things in a more controlled manner. He went ahead and he synthesized or he purified each of the amino acids that were known about, and then he was able to mix and match them in order to figure out things like how much of each of them do you need, and well, what is missing. He took what was known about the amino acid content of casein, which is a major protein in milk, and he modeled that. He took the same amount of each of the amino acids that's in casein and mixed them together to get this kind of artificial casein. But even though the casein was good enough, so at least if you added histidine, the mice, when you gave them casein, could survive on that. The synthetic mix wasn't. Even if you added histidine, they couldn't make do. So he knew that he was missing something in his synthetic recreation. There was something else in that casein that he was missing, another amino acid. And so he wanted to figure this out. Here you can see the results of some experiments he did in rats, where he gave them the mixture that he made. And you can see that the rats, this is showing their weight. And so their weight was declining over days because they were missing some sort of essential nutrient. And you can see that quantified here as well. So Rose set out to find what he called the new essential, starting with 40 kilograms of a different protein fibrin and doing a lot of stuff. And so I'm just going to I just highlighted some of the words talking about all the different things he did. But he extracted 17 times with 40-liter portions of butyl alcohol. That is insane. I can't even imagine how they dealt with all of that waste, but I'm not going to think about that right now. That was a lot of work. And then you have a ton of liquid. So he concentrated that, cooled it, filtered it, did several washes. And then he got these kind of like different extracts, and he tested those different extracts. And he went through all this work on this one extract, and he resuspended it, and he mixed it, and he filtered it. And then he re-extracted it 3 times with 1.5 liter portions of that butyl alcohol and 5 liters of solvent. And he got 56.4 grams of something, But that actually wasn't what he was looking for. It didn't have the activity he was looking for, and so he discarded it after all that. But he still had another portion. And so he took that other portion, and then he took that other portion and he tried a bunch of stuff that was unsuccessful. Poor guy. You can like almost hear the pain in his voice. Insomuch as the measures employed were not satisfactory, they need not be described. So what he ended up doing it was dissolving it in acid and then treating it with acid and filtering it and washing it and evaporating and filtering it and adding stuff, precipitating it, washing it, filtering it, adding more stuff, concentrating it, fractionally precipitating it, and woof. But he started seeing the separation of little needle-shaped crystals. And so he saw something like this. And this is what he saw when he was starting to get it pure. And he said that it looks like the crystals that you would get with tyrosine, but it didn't respond to the chemical tests that the same way as tyrosine would. And this wasn't pure enough. He did more and more pure crystallizations, getting rid of more and more contaminants, and he got these pure crystals. He didn't have much of those crystals, so he only had 4.5 grams from the 123 grams of crude material that came from the 40 kilograms of the fibrin protein, so about 0.8 grams per kilo of fibrin. Oh man, can't even imagine. So he tried a couple of different methods to try to get stuff better, but ultimately no procedure has been found, which is not exceedingly laborious and which does not involve a considerable number of recrystallizations after the substance has been freed of a large proportion of its contaminants. So basically, when you're doing these recrystallizations, you are kind of pushing out more gunk. And there, when you get it really, really pure, there's nothing else in that crystal except for the thing you want. If you have a pure crystal, it's all what you want. And so this is all what you want. And he barely had any of it, but it was still enough to test. And in fact, when he gave it to the rats, now instead of dying, the rats were able to survive. So he gave them the same mixture as before, but now he added in this new thing, this new essential he had found, and voila, the rats were able to grow. They wanted to characterize this new essential, and so they did a number of different chemical tests on it, figure out how much of each element it had, find out how much of each, find out how many, find out like its chemical composition in terms of the makeup of its different atoms, find out the way that it rotates light, find out its melting temperature, things like this. Do a variety of chemical tests to try to figure out what it was. They were able to identify it. Partly, they found that it was one of the 4 optically isomeric alpha amino, beta hydroxy, and butyric acids. But they couldn't figure out the configuration around the beta carbon atom. So basically, if you look at the structure of threonine, you'll see that you have 2 places where you have what we call steric centers. Stereochemistry refers to the direction in which things are sticking in space. So for all of our amino acids, except for glycine, they have at least one stereocenter. The alpha carbon, where the side chain's sticking off, one side chain is sticking off, and the carbon is also attached to a hydrogen. And in the case of glycine, you have 2 hydrogens. So that's why glycine isn't chiral, because in order to have a chiral center, you need to have a carbon attached to 4 different things. With all the other amino acids, however, because their side chain isn't just a hydrogen, you do have chirality. So this carbon is attached to the carboxylate, it's attached to the amino group, and it's attached to the side chain as well as to a hydrogen. If you think about things in one orientation, the side chain can kind of be sticking forward or backwards, and that would be akin to L and D forms. Our bodies use the L forms, and so all the amino acids are basically going to have that D or L, except for glycine. But most of the amino acids are only going to have that one stereocenter. If, however, we look at the structure of threonine, we see that here we actually have another stereocenter. This carbon is not attached to 4 different things, but this one is. These are all different, and so threonine is going to have the second stereocenter. When they did their experiments, they were able to determine that it was L, but they didn't know which way this is facing. So you can think of like, is that oxygen facing at you or is it facing back? However you want to think about it, there are different combinations that can be happening. He was able to figure out the L part, but he wasn't actually able to figure out what direction this one is. And so that was going to be the subject of future work in order to figure that out. H.E. Carter synthesized all four of the potential options and showed that the natural one corresponded to 2S, 3R, 2-amino, 3-hydroxybutanoic acid. Whoosh! And so as to all that naming, basically if we think of that, this was our alpha carbon, and we say that it has an alpha amino group as well as an alpha carboxyl group. But because you're naming in terms of the acid here, so this would be your alpha amino group. And then this would be beta hydroxy. And so this would, if this is the alpha carbon, this is the beta carbon, and it's got a hydroxy group. And then butyric acid. So but, that's like 4. And so 4 carbons, 1, 2, 3, 4 carbons. We have a carboxylic group, we have an amino group, and we have the beta hydroxy group. And voila, that's how we get the name. And then in order to figure out the direction, then you had to go and synthesize the different options and then run the tests on each of them to see which of them behaved like the one that he had isolated. And in case you were wondering, isoleucine is the only other one with another chiral center with these 2L stereoisomers, because all the other ones, most of them have like a carbon that's attached to 2 hydrogens and something else, or 3 hydrogens and something else. So that part wouldn't be chiral. But with isoleucine, you can see that we have this kind of central carbon here as well. In the case of valine, here you have the same group sticking out here. So this is not chiral. But this one, we have kind of 2 carbons on this side and 1 carbon on that side and a hydrogen and then that whole thing. Yeah, that is going to be a chiral center, just like we had that chiral center with our threonine, but not with our valine and not with any of the other amino acids. And so that complicated them finding the actual full structure of threonine. But they were able to show that it was that new essential that he'd been endeavoring to isolate for several years. And for the first time, they were able to induce growth in animals, giving them only a diet of purified amino acids. So like synthetic mixture of highly purified amino acids, as opposed to just giving them proteins. As they write in the paper, it is our purpose also to determine the quantitative needs of the organism for each nitrogenous essential. Eventually, such data might provide a new and final answer to the question of the protein requirements of man and animals. And so he started with the animals. After doing the rats, he moved on to dogs. But dogs still aren't people, so if he wanted to know protein requirements in men, he needed to find some men. And one of the places scientists often go, especially in the olden days, when you wanted to do an experiment on some people, would be students. In this case, Rose turned to some graduate students, healthy male graduate students, and he fed them a diet in which he gave them all the nutrients they need. So cornstarch, sucrose, butterfat without protein, corn oil, inorganic salts, known vitamins. So everything other than the amino acids that they would need, and then mixtures of highly purified amino acids. So we could tell which ones they need. Yeah, also, side note, they also had a large brown quote-unquote candy, which contained a concentrated liver extract to supply unknown vitamins, sugar, and peppermint oil to provide a never-to-be-forgotten taste. Yeah, I doubt they forgot that. They might also not have forgotten the kind of irritability they felt when some of the amino acids were withheld. So isoleucine, leucine, tryptophan, lysine, methionine, phenylalanine, threonine, and valine. Yep, they're threonine. Rose was able to show in these volunteers that removing the threonine actually led to differences in the amino acid, differences in the nitrogen levels in their waste, which was how he was measuring whether or not they were essential rather than just like waiting for them to lose weight. And don't worry, he was able to give them the amino acids and they perked right up again. Although there was likely a slight panic among some of the volunteers when another lab reported that threonine deficiency lowered sperm count. But this theory was later counted out, so phew for them. Anyway, we have them to thank for a lot of this, especially because all they did it for was free food. Yeah, there's way easier ways to get free food, especially in grad school. There's like free food everywhere. But anyway, they got $1 a day and the prospect of getting their initials in print in Rose's widely read publications. So probably when I get past the IRB, so the Institutional Review Board, as is these days. But it was an important experiment, and hopefully people who do experiments with their students will pay them better. So thank you to the grad students, as well as thank you to William Rose, R.H. McCoy, C.E. Meyer, for their work showing that, well, threonine was threonine and showing that it was essential. So there you have it, some cool threonine biochemistry.